welcome everybody to the, can you hear me well, by the way? Do I sound good? Perfect.
We can hear you.
So welcome to the AARO seminar series centered on the topic of auditory cortex and thalamus. Today, we're delighted to have Dr. Jonathan Simon as our speaker. My name is Maria Chait. I'm a professor of auditory cognitive neuroscience at UCL in London. And I'm also the chair of the AARO membership committee, and I will be the moderator for today's webinar. And I'm seated in a seminar room at the UCL Ear Institute where a bunch of my colleagues are going to be watching the talk with me. So before starting, there are a few housekeeping things. So during the presentation, if you have any questions, please use the chat functions to avoid interrupting the presenter. Following the presentation, we'll go over those questions, and in addition, you'll be able to unmute yourself and ask questions live. Any questions that will not be addressed due to lack of time will be answered by Jonathan in a follow-up email. And there's closed captioning for this meeting. It's available for you to use, and the session is also being recorded and will be made available for viewing offline later. And if you're having any technical difficulties, please email AARO. I understand that they're going to put the email on the chat now for assistance. So a little bit of background on Jonathan. Jonathan earned his bachelor's degree in physics from Princeton University and his doctorate also in physics from the University of California, Santa Barbara. And he did postdoctoral research in theoretical general relativity in Wisconsin and at the University of Maryland in College Park, where he has, among his highly cited publications in that domain, is my favorite, Black Hole Thermodynamics and Lovelock Gravity, which I encourage you to check. But then luckily for all of us, in Maryland, he embraced the field of neuroscience or auditory neuroscience. He joined the University of Maryland's Electrical and Computer Engineering Department as faculty in 2001, the Biology Department in 2002, and the Institute of Systems Research in 2013. He's the co-director of the KIT UMD MEG Center, and also the director of the Computational Auditory Neural Systems Laboratory in Maryland. And he is currently the PI on an NIH R01 grant, which is titled A Multi-Level Auditory Processing of Continuous Speech from Acoustics to Language. His expertise is in applied and theoretical neuroscience with an emphasis on auditory neuroscience. And his main research focuses on neural processing in the auditory system. From specialized processing that's found only in humans, for example, speech processing, to generalized processing found in most mammals, like auditory attention and localization. And another of his research areas is the investigation of the effects of aging on auditory processing, especially with respect to understanding speech and noise. Jonathan occupies kind of a unique position in our field because he's able to apply his expertise from physics and signal processing to unravel the complexities of hearing and listening. And his pioneering work, which I think he'll be talking about today, which showed back around maybe 10 years ago that brain activity is decodable from the listening brain. It has driven kind of fundamental shifts in our understanding of hearing over the past decade. And on a personal note, Jonathan is one of the kindest and nicest and most approachable members of our community. And if you haven't yet, I strongly encourage you to take the opportunity to chat to him at the next opportunity. And for me, it's a special privilege to be introducing him today because I began my career as a neuroscientist in his lab many years ago. I was a PhD student, one of the first PhD students in his lab. And under Jonathan's guidance, I learned to be meticulous, to select the right tools to use for my work really carefully, and to ensure they understand why I'm using them, what the purpose is, and what artifacts they can generate in my signal. And that's a really important lesson to have learned. And more importantly, I think I learned to be curious and inquisitive. And I try to apply these in my career to this day. So without further ado, it's my utmost pleasure to welcome Jonathan, who will be presenting his work titled Neural Representations of Same Species Vocalizations in a Human Primate Model. So on to you, Jonathan.
Maria, thank you so much. That was very kind of you. And I have to say that when you left Maryland, you left some awfully big shoes for people to try to fill. And all of my students and postdocs have been encouraged to try to fill them as much as well as they can. So thank you. It's great for you to be here. Title of my talk. You might notice that there's a little bit funniness going on there. Human primate model. Let me see if I can just make sure that, yes, okay, so human primate model. Why didn't I just say humans? If we're talking about humans, same species vocalizations, why didn't I just say speech? Well, that was my original title. But I wanted to make sure that in this series of thalamus and auditory thalamus and cortex that I was not being so focused on humans. There's a lot of other animals that have a thalamus and a cortex used for auditory processing. So I wanted to try and be as general as I could. So this is the actual title, and I will try and cross bridges whenever I get a chance. In case I get rushed at the end, which I usually do, I wanted to thank the current members and affiliates of my lab, faculty collaborators, and especially agencies that have provide funding and support for this work, including the NIH, the NSF, and DARPA and universities. I'm also, as the first speaker in this series, encouraged to mention the entire series, and I would certainly like to encourage people to come to Dr. Laurie Holtz and Dr. Loss Williamson's seminars later on. All right, so an outline of what I'm going to talk about is I want to focus on auditory neurophysiology in animals versus non-invasive neural recordings in humans, and where there is common ground between them, because as techniques, they're quite different, but we're typically asking the same questions. Here, when I say human recordings, I mean EEG and MEG. I won't be covering ECOG, which is highly complementary to both of these, but does not replace either, or hemodynamic techniques. I'm also going to emphasize the neural processing of same species vocalizations and the neural processing of speech as potentially the same thing as they are in humans, but as different things when the experiments are carried out in animals. And I want to keep in mind that speech, while it might be special, is also a vocalization that is a carrier for language, but it can be treated just as a vocalization as well. And I'll also be talking a lot about categorical perception, but even more so about the neural processing, categorical neural processing. So the neural processing underlying categorical perception of elements of vocalization / speech. So before I get to thalamus, let me talk about the brainstem. On the left is a picture, a nice picture of the auditory system. I'm supposed to be providing a broad outline of how the auditory system works, where you can see there's many stages. This part of this figure only goes up to the inferior colliculus. And on the right is a typical neural response profile from the auditory brainstem conducted in human research, and looks quite different, but conveys much of the same information, certainly not all or even most, but much of the important information. And let me go through how that even makes sense. This is called the auditory brainstem response, or ABR. It's typically averaged responses to some sort of punctate stimulus, like a click train or tone train. It's characterized by three robust peaks, here labeled 1, 3, and 5. And people who have looked at the origin of these responses, of these peaks, show that wave 1 is dominantly from the cochlear nerve or auditory nerve. Wave 3 is from the cochlear nucleus, which covers a lot of ground there, but auditory localization is hard for such a anatomically small area. And wave 5 is dominantly from the inferior colliculus. And so one of the differences between these non-invasive recordings in humans and invasive recordings in animals is, even though we lose a lot of information about where specifically things are going on in specific cell types, we can see multiple areas at the same time, just in one view. And I would consider that a blessing, although there's also curses. The responses to speech in the brainstem in humans also look quite similar after a bit of signal processing. On the right, we have the analogous responses not to clicks, but to speech and to music. And when processed in a reasonable way, called the temporal response function, or TRF formalism, we also get these same three peaks. I'll talk a little bit more about TRFs, but essentially, these TRFs, or responses, are obtained by deconvolution, a long continuous stimulus with a long continuous neural response. And it also matters how we're deconvolving. And here, this is with a stimulus representation that comes from an auditory nerve model from Laurel Carney's lab. And the most important thing here, I'd say, is it's still characterized by these same three robust peaks that would still be generated from the same original locations. And this comes from Ross Maddox's lab. Temporal response functions are a model, essentially, of how stimuli can generate responses. A typical cartoon TRF here is in red, surrounded by a green circle. Okay, I got my mouse. And it's a way of mapping a stimulus, in this case a punctate stimulus, into a response. And if there's only one isolated stimulus, then you get responses. If you get a pair of isolated stimuli, you get a pair of responses. But if the two stimuli are very close to each other, then the TRF is a linear model, and the individual response components add, so that the actual predicted response is a linear combination of the two. And furthermore, if the stimulus changes either in amplitude or even in sign, then because it's a linear model, the response also changes commensurately in amplitude and sign. And one of the more important things is that this can be applied even to continuous stimuli, where you just essentially break up the stimulus into little bits of little amplitudes at different times, and you linearly add them at each time. And so it works just fine for continuous stimuli and continuous responses. And of course, we don't know what a TRF might be as a model. We wanted to estimate it from the stimulus and response. And the way we do that, I won't go into details here, but there's a nice literature on that. This was first done by Ed Lawler, and also done around the same time by my then student Nai Ding and myself. And estimating the TRF from the stimulus and response gives you the TRF, but also gives you a model fit. Because you get a predicted response that you can compare with the actual response. So at any rate, going back to the auditory brainstem response, we can see that even though we can use continuous speech as a stimulus, not just clicks, we still have much information, much of the same information. And then we can compare that to what we think is going on in the rest of the auditory system, given other knowledge from animal studies, but also we can just compare them in parallel in general. Making our way up to the thalamus, because that's one of the topics of this series. If you do use the same technique, and this is also from Ross Maddox's lab, if you look at responses that are a little bit later than the brainstem response, you get what are called middle latency responses, MLRs, and those originate from the thalamus. But these responses are also generated from the TRF formalism, and this means that we can get from continuous speech brainstem responses and thalamic responses, and even later cortical responses. Ross's lab is not the only lab that does this. If you got to see Carla Root's poster from my lab at ARO, you would have seen that we are now attempting similar things with various stimuli, including cocktail party stimuli. And in my lab, we've also looked at thalamic responses from a totally different point of view. It turns out that if you look at TRFs for cortical responses, but in a particular band in the 70 to 100 hertz range, so about 80 hertz, then you can see TRFs that have a pretty short latency. They're about 40 milliseconds. That's pretty short for human cortical responses. That indicates that it's primary auditory cortex from a latency perspective. If you try to localize that, it also comes from primary auditory cortex. But the reason I grouped this with thalamus is because these are now MEG responses instead of the EEG responses we saw before. The source currents that generate MEG and EEG responses are dendritic currents, meaning they're reflecting the input to the neurons rather than the output. And the input to primary auditory cortical neurons comes from both the thalamus and cortex. And so really what we're seeing here are thalamocortical processing. And that also is consistent with this being in the 80 hertz range. 80 spikes responding to 80 hertz stimuli is awfully fast for most cortical neurons, but it's not so fast for thalamic neurons. And then a quick shout out that this same technique can be used to be seen selective attention effects in the same thalamocortical response. Okay, now proceeding, I want to talk more about speech specifically, but also talk about vocalizations that aren't necessarily human speech, and emphasize, as I said before, that speech is a vocalization that's also a carrier for language. And then at the same time, talk about the neural categorical neural processing, which is what underlies categorical perception. So a nice little example of categorical perception is if you have the sound bad and the sound dad, and one linearly morphs bad into dad, that subjects will not observe that there is a continuum. So it won't be a linear discrimination, but it will be this sigmoidal steep in the middle discrimination, where the first few elements are ascribed to bad, and the last few are ascribed to dad, with very few around 50%. Now, what's especially interesting about this particular data set is that this doesn't come from people. This comes from rhesus monkeys. This is an experiment from Yale Cohen's lab. And so even though speech vocalizations are not same species vocalizations for rhesus, they still perceive this categorically. And not only is it their perception, how they report behaviorally. Oh, I'm skipping. I shouldn't have said that. We'll come back to that later. I want to talk more about categorical perception first. For one thing, why do we have categorical perception? Certainly, it adds robustness communications. It's something analogous to why digital communications is sometimes superior to analog communications in terms of robustness to noise and poor conditions. But it also means that categorical perception is really a robust percept. And here I have 4 spectrograms of each of them has 4 phonemes. But the four phonemes in these different contexts, spoken by the same speaker here, by the way, look quite different. The four phonemes here in each of these are an initial H, followed by an and then an L, and then a P. And so they all spell out, they all correspond to the word help, but for our purposes, they each of these has a phoneme H, which is acoustically different in every case, and has a phoneme L, which is acoustically different in every case. So it's really the phoneme that we're going after here, not the sound. And so we have four different versions of the word help. I'm throwing this in because won't you please help me, but I can't think of any other reason to throw it in, but it just popped into my mind. All right, now we're going back to these this categorical perception of these rhesus monkeys that were in Yale Cohen's lab. And here he showed that it's not just the perception, but cortical neurons in the anterolateral belt, the ALB, the neurons themselves respond categorically to these phonemes. So it's, of course, one would hope that the percept is related to what's going on in the brain, but here they were actually able to show some neurons that underlie this perception. In humans, it would be nice to be able to measure responses to phonemes. We have the issue that in humans, if we're doing non-invasive recordings, we have to separate out the responses to phonemes from the responses to the sounds of those phonemes. And this turns out to be not so hard. We just do multivariable regression in the time domain, which is a complicated way of saying we're going to do multivariable TRFs or MTRFs. And that basically amounts to we're going to use several different representations of the stimulus, estimate several different TRFs, one for each of them, and those will correspond, the sum of those from different regions of the brain will correspond to a mixture of neural signals, and we will estimate in a way that gets us the best TRFs to most closely mostly correspond to the actual responses we see. These are pretty straightforward to do if you want to try them yourself using either the MTRF toolbox from Ed Lawler's lab or EelBrain, which is written by Christian Brodbeck, a former member of my lab. That's not quite enough because phonemes, while not identical to their underlying acoustics, are still pretty strongly correlated with them. And even MTRFs have trouble when the predictors are too correlated. So in my lab, we've gone to using measures that are less correlated with the acoustics. And to do that, we use phoneme-based predictors that are based on linguistic statistical distributions, specifically phoneme surprisal and phoneme cohort entropy. These are both phoneme processing measures, but that are not correlated with the underlying acoustics. And a bonus of using these is that we might learn something about the neural processing of these linguistic measures. Let me tell you what these measures are. Phoneme surprisal is, in a word, how surprising a given phoneme is given the previous phonemes that the listener has heard already. So for instance, if one is in the middle of processing a word and the word begins with the sound K, if the next phoneme is an coming from the letter M, that's not too surprising. 45% of words in a large database of English language use, if they start with the sound K, are followed by the phoneme corresponding to the letter M. And examples of that are came, which really makes up most of them, but it could have also been Cambridge. Whereas if the next phoneme were the S sound, that's still pretty not surprising, pretty common. Could be case, could be cases, other words. Whereas if we go down to the phoneme cake is much less common and would be more surprising and would be even more surprising. And so we get this hierarchy of frequency, and 1 over frequency is termed surprisal. So basically our measure is 1 over frequency, although we do also take the the log base 2 of it to turn it into bits as a convenience. So phoneme surprisal is one measure we use. Cohort entropy is another. It's also based on statistics, but it's a different set of statistics. It's how unpredictable the current word is. And so, for instance, if we started off with the sound lay as the beginning of our word, and then we heard the next phoneme correspond to the letter K, so we got lake, it's very likely, based on the English language, that lake would be the entire word, and we were done with it. Although there's a 5% probability that that we wouldn't be done with it, and that we would be listening to the not yet completed word lakes. Cake is similar, where cakes is more common relatively than lakes is, but caked is also an option. In contrast, if we started off with bay and ended up with a ke, then it could be baker, bacon, baked, bake, and these form a spectrum of entropy. This is a very low entropy set of possibilities here. There's a very high entropy set of possibilities here. And this entropy is the measure that we use. So we'll do this MTRF decomposition of the responses into acoustic features and to phonemic features in this way. And let me tell you a little bit about the study design here. We used 4 different kinds of speech-like sounds. The speech envelope modulated noise, which is based on speech, but all of the intelligibility has been taken away. The carrier has been replaced by pure noise. And so this should sound like it was based on speech and have the same rhythm of speech, but it should be completely unintelligible. Whereas the speech that was based on, which has exactly the same rhythm, is highly intelligible.
If you happen to find yourself on the banks of the Ohio River on a particular afternoon in the spring of 1806.
And just to avoid certain confounds, we also use 2 other stimuli. One of them is the stimulus of non-words or pseudo-words.
Sustament evilis, Joserval edfolk provericant zin tehovasybe by consin skeding pitablion glad apres prianus.
That sounds a little eerie to many people, because it sounds like English, but it is not English. All the phonemes are English, but there are no words. And then also, as another control, we use the same we use the same words that came from the narrative, although from a different section of the narrative, so the words were not presented earlier, but it's completely scrambled their order.
A liquid is only speak, second even for good reach the attack us. Living fact, which it was plants.
Which to me sounds like I've gone to a seminar, but in the wrong department. I understand all the words, I just don't understand them in that order. So we compute the TRFs from all these different kinds of stimuli. First, we compute the ones that correspond to acoustics. These, I'm going to use the acoustic envelope onset as one predictor, acoustic envelope as another. People who only use the acoustic envelope and calculate TRFs, this is often called speech tracking. We're trying to be more complete by using both the envelope and the envelope onsets. In fact, the envelope onsets, if you look at these units, give stronger, more robust response profiles than just the envelope itself. And you can see that the noise gives a TRF which corresponds to a less activity than the other word or word-based stimuli do. Also, both of them have a very very standard profile. These are all plotted as power, so I don't have the sign of the currents here, the neural currents, but I put them here with little plus and minus signs. This is a positive polarity. This is a negative polarity, and this corresponds roughly to the P1 response of EEG to a punctate sound, and the N1 response of EEG to a punctate sound. And that's true for both the envelope onsets and the envelope. And we get these responses at about 60 milliseconds for the first positive polarity response, and about 120 milliseconds for the second one, which is also acoustic, but like the N1 or the N100 in MEG, this is highly attention dependent, and especially selective attention dependent. I want to bring back my listeners, my visitors here, attendees who care about animal work. You may be thinking 60 milliseconds, 120 milliseconds, that's not the kind of latency that I see in my neural responses in individual cells. 120 milliseconds is not as crazy late as it might seem. What I have here are STRFs based from recordings of ferret A1 listening to speech, to human speech. But these are based not on the spike responses, but based on the LFP responses. And EEG and MEG responses are, because they are related to the dendritic currents primarily, are more related to LFPs as measured in animal neurophysiology experiments. And these STRFs are really giving routinely latencies that are over 100 milliseconds. So as long as the whole brain is responding and processing these sounds, having some aspect that's coming at 60 milliseconds or 120 milliseconds isn't crazy even in animals. Going on to the phonemic responses, there are really nice TRFs that correspond to phoneme processing, phoneme surprisal processing, and the phoneme cohort entropy processing. I'm going to not concentrate too much on the non-word because those are a little funny. This cohort entropy, you'll recall, is based on when the word started. And when a word starts, in the case where there are no words, none of the words are words, is ambiguous, and it's clear that the brain is having trouble processing what a word is and what a word isn't when none of them are actually words. But if we focus on the scrambled speech and the narrative speech, there's in all of these cases a low-level phoneme processing peak at around 80 milliseconds, which is really not that much longer than the 60 millisecond acoustic peak. And then in both of these more complex responses, there's a later negative peak at around 350, 400 milliseconds with a negative polarity. And this is a lot like what's called an N400 response to speech with semantic content. And it's associated with predictive coding in those cases. And so one is tempted to interpret these responses as, first of all, evidence of categorical neural processing, because these are phoneme responses that are divorced from the acoustics, and phonemes are categorical. But also there's these two different stages. The 80 milliseconds is so early that that's consistent with predictive coding models. This is a response not actually based on what is experienced by the brain, but what is likely to be to have been experienced by the brain. And then this later one, this negative N400 like, is associated with error correction in predictive coding. I'd also like to go beyond phonemes. In human speech, phonemes are the building blocks of words. Words and groups of words are used to convey meaning. And so a lot of the reason we use, maybe even almost all the reasons we use speech are to convey meaning. But animal vocalizations, non-human animal vocalizations, are often used to convey meaning. And I want to take another study from Yale Cohen's lab, where they looked at rhesus monkeys, where some of their vocalizations transmit information regarding the quality of food that's presented to them. There's a particular vocalization called a grunt that means the food is of low quality. And there's two very acoustically distinct vocalizations that indicate the food presented in front of them is high quality. Um, in this experiment, Gifford et al. From Yale's lab, they were able to show that in one region in the rhesus brain, in ventral prefrontal cortex, VPFC, that there are neurons that respond categorically, and here it's based on the meaning, not on the acoustics, and certainly not on phonemes, because there's no human speech here. These are rhesus vocalizations. These are neurons that encode transitions between calls of different categories, and they do not encode transitions between acoustically distinct but semantically the same vocalizations. But they do they do encode transitions when the sounds are different, but the meaning is the same. Going on to the human case, and words do often convey meaning in human speech. We can extend our MTRFs, our multiple TRFs, by adding word level features in addition to the phoneme features. I'll tell you about the 3 word level features that we use. Word onsets, you might think that word onsets are strongly related to the acoustics, and they are, but we're already trying to control for acoustics, and they're not that tightly related, because here's a great example from Norris and McQueen. The phrase, the catalog in a library, why the brain is interpreting this in real time could be the catalog in it could be the catalogin, the catalog inner. It's possible to try and guess what the word boundaries are in real time, but it's also impossible to get it right sometimes until you have enough context to go back. And so we might even expect that the TRFs here also have an early component and a late component. Word surprisal is analogous to phoneme surprisal. We use 2 different kinds. Without context, is we just take how common the word is in this same database of word occurrences. The most common words like the, to, and are very frequent. Again, we take 1 over that to be surprisal, and then take the log base 2 to turn it into bits. And then we do a more interesting one. We have a contextual word surprisal, where if you're listening to, for instance, the phrase, if you happen to find, then using a large language model, in this case, it's a GPT model, we can look at what the probabilities of the next word is, whereas yourself is very probable, very unsurprising, but we can go down and list all of the potential words that might be more surprising, and give each of those a surprisal value as well. So when we look at the word responses in humans, we can again see there's clear evidence of these word-driven responses. Here's the onset. Here's the surprisal without context. This is, again, categorical neural processing, because we've taken out the acoustics. And again, we see that there's an early peak. This case is about 100 milliseconds, which again is not that much later than the phoneme early response at 80 milliseconds. And again, an additional N400-like set of processing stage, which is indicative of predictive coding. And if we ask about surprisal without context, compared to surprisal in context or contextual, the response to the word surprisal that's contextual is actually about twice as big as the word surprisal without context. They even seem to have very similar dynamics. And so one interpretation of this is we're really seeing what's going on, mostly from the contextual word surprisal that's contributing more to the response. And in fact, maybe this is not really contributing much at all, except that our version of word surprisal may not be the same as this particular human brain's version. And so this might just be catching a little extra residual. So from this stage, we've seen that there are cortical responses that are time locking to these emergent features, from acoustics all the way to contextual word surprisal. We've seen phonemic and word-based cortical processing that is categorical, like those two rhesus experiments that we saw. Though the rhesus experiments, the phonemic one was categorical, the second one was not word-based, it was vocalization-based. I'd also like to point out that contextual word surprisal is not unrelated to semantics. I certainly would never say that it's the same as semantics. I would not say that a large language model understands semantics. But in the human brain, because they are correlated, one shouldn't be surprised if the contextual word surprisal response is related to a semantic response if we were able to separate those out, if we were able to come up with different ways of separating them out. And then also, I think this is extremely interesting and potentially of very high importance. These long latency stages are in line with predictive processing models. And then let me see. Oh yeah, so I also want to talk about an application of what we've just seen of these basically of the categorical word processing. And the application is, can we tell when or if distorted speech is intelligible or not? Because it's true that even very clear speech can be unintelligible if it's not in a language that you understand, for instance. But much more common is that very distorted speech may or may not be intelligible. But what's important is that very distorted speech can still be intelligible. And if you have a hearing disability, but you use some sort of device or hearing aid to lessen the distortion, then it would be really nice to know whether that the person listening to this perceives the speech as intelligible or not, without necessarily having to ask them. And so I turn this into the question of, can we use these neural categorical encoding of speech features to determine when the brain processes speech as intelligible or not? This is the design. We use 3-band noise vocoded speech to make it extremely unintelligible. So for instance, here's an example of highly unintelligible speech. And then we play what the original undistorted speech was, which is of course highly intelligible.
Slice an apple through at its equator and you will find 5 small chambers arrayed in a perfectly symmetrical starburst, a pentagram.
And now what's extremely interesting is because your brain has been primed, if you listen to the original unintelligible vocoded speech again, it now, for many people, will appear intelligible. And so we did this on a whole bunch of subjects. We inquired about their perception, and we showed that if we compared the pre-clear stimulus with the post-clear stimulus, that their reported speech clarity increased from pre to post. And more interesting from the perspective of this talk is we also looked at their word onset TRF, which, as before, shows that there's typically an early stage and a late stage. And here I have the TRF with the polarities actually expressed so that positive means positive polarity and negative means negative polarity. And if we look at over the subjects, the amplitude of their early peak between pre and post, the robustness of the response, the amplitude, increases significantly from pre to post. This is only in the left hemisphere, but that's not surprising given the language aspects of this processing. And this is a moderate effect size. But even more gratifying is that if we look at the late, the increase there is not just moderate, but it's a large effect size. And the significance has changed from 1 star to 3 stars, if that floats your boat. But more importantly, I think, is that the ratio of this peak to this peak has increased while the error bars have gone down. So that's the large effect size. And I find it especially gratifying that that this is happening more in the late error correction stage of predictive processing, because from our perception point of view, we don't care whether it's early or late. We don't care whether our brain figured something out in 100 milliseconds or 400 milliseconds, as long as our percept is all continuous and in real time. And it makes sense to me that this kind of priming, this giving the auditory brain access to other information, would occur at a later stage rather than an earlier stage. Very quickly, if we look at the acoustic responses instead of these word-based responses, there is no change. Change is really only seen in the word-based responses. It's not shown here, but if we look at the word surprisal response, that also increases. And that means what we have here from an application point of view is that these neural word-based responses might perform the role of an objective measure of intelligibility. That if we really want to come up with a system where we can automatically tell whether a certain set of settings on a hearing device increases in intelligibility or not, we don't need to stop the subject after every sentence and ask them what the words they heard was and score them behaviorally. We have this possibility of just recording their MEG or their EEG and then automatically come up with a small set of settings that all seem to improve intelligibility, and then an audiologist and the patient can work together to choose which one is the most satisfactory. So in summary, because that's the end, I've been talking about investigating neural speech and language processing in humans for the most part. This, as I just mentioned, was at the end, does have potential clinical applications. But of course, humans listening to speech is a form of animals listening to listening to communications, to vocalizations. And so one can use humans as a model, as an animal model for what happens in general primate or general mammalian brains. And we might even be able to talk about questions that can address how language evolved. Human speech is special, certainly in a neuroethology sense, just like barn owls have better spatial hearing than other birds, but their general auditory processing is not that much different than other birds. They just have a specialization. Similarly, our speech has a specialization, and it did give us language, which is arguably unique to humans. But maybe we can start talking about connections. And then at a more down-to-earth level, talking about categorical neural processing, the neural processing that underlies categorical perception in vocalizations or speech or both. We saw that for speech, we saw that at the phoneme level and at the word level, and that this is dissociable from acoustics. It can, for humans, provide this with new insight in the linguistic domain, and my lab has contributed to some of that. And then we also saw from an applications perspective, these categorical neural processing responses aren't available if the speech is unintelligible, whereas they are available when the speech is intelligible. And that lets us see much more clearly in terms of the hierarchy of what's going on in auditory processing, where one stage begins and one stage ends. Thank you very much. And I will now encourage Maria to take questions.
Thank you very much, Jonathan. And yes, please, so to remind everybody, you can ask questions in the chat. You can ask questions by raising your hand and unmuting yourself. whilst people think about their questions, I'll ask kind of a general one, which relates to the tool that you're using, the TRF. You mentioned, Jonathan, that it's like a main sort of assumption behind the TRF is that it's linear. It's assuming linearity. And you've shown, the various things that you can use the TRF for. But is the assumption of linearity fair, do you think? And if not, is there anything that this assumption is kind of precluding us from seeing or might be misrepresenting about
Sure.
brain activity?
Yeah, sure. That's a fantastic question. Because the brain is not linear, and I'm using a linear technique. So the question almost begs itself, and I appreciate it coming out first. For two parts to the answer. One is that if there are any linear aspects, if there is if a sound or aspect of a sound is being processed non-linearly, but one can break that processing up into a linear piece plus a quadratic piece plus a cubic piece, etc., then we still have access to that linear piece. So even though we may not be as good as we could be at predicting the response, we're still getting some fundamental aspect of it. And I would use the analogy there of using pure tones to look at tonotopy in the auditory brain. Most sounds are not pure tones, but the brain is laid out tonotopically. We can get a lot of information from using only pure tones, and it's extremely helpful. It's not the be all and the end all, but we still look at tuning curves. The other answer is that it's when we say it's linear, what do we mean linear with respect to? And part of the science of doing this analysis is finding neural representations that we can get linear responses to. So we're not looking at the raw acoustics here. We're looking at envelopes or envelope onsets or phonemes. We're looking at higher level, very non-linear aspects of the sound that we then can look for linear responses to or linear contributions to. And so that also helps in the sense of finding things that the linear representation is robust, even though it's not the be all and the end all.
Well, thank you. Brian, do you want to ask your question? Or shall I ask it for you? So Brian is asking whether you have data. Oh, there's Brian. Go on, Brian.
Yeah, I really appreciate the level of measures that you're using here in your descriptions, and I know you've done work with aging, and I'm wondering if you have data on how these measures, or do they give us some insight into what's happening in the aging brain when we have more difficulty understanding speech?
Yeah, that's a great question, and we do. The elephant in the room, as far as the answer to that, is that the TRFs in aging brains tend to be larger than in younger brains rather than smaller, which seems counterintuitive. There's probably several reasons for that. The biggest reason is likely a change in excitation inhibition balance. The aging brains, and there's a lot of evidence for this in animal models, have less inhibition relative to excitation. And so because we're seeing the currents not being canceled out so much by inhibition, we see larger currents. And so therefore we see bigger TRFs. But that doesn't mean that the older subjects hear better or understand speech better, because there's a lot of evidence that they don't, or that we don't. Because inhibition plays an important role in shaping how sound is processed by the brain. And so this larger TRF doesn't indicate better hearing, better understanding, or better auditory processing. As far as the other measures, those don't seem to be larger or smaller dramatically. Some of them are a little smaller. we're especially interested and looking at right now is what happens to those same measures when older subjects have undergone some auditory training, some listening to speech and noise training. Can we change what these TRFs look like from training? We know that we can change how well they do in this listening to speech and noise task. We believe that's because their brain has changed, and we found some nice evidence that their brain has changed due to this training. Thanks.
Thank you. Chait, do you want me to ask you a question, or do you want to unmute and ask it yourself? So can't see Kate, so I'll ask for her. So Kate says, you mentioned that humans have phonemes while animals do not. Is there any way to break down vocalizations among other species in a comparable way to how we do in humans? And if so, do you think it would make a difference in their response?
So I am far from the expert in that, but certainly yes, there's a lot of structure to some animal vocalizations, bird vocalizations in particular. I know that there is a lot of work in looking at how, but at the hierarchical structure of bird vocalizations. Question, and part of the question, do I think would make a difference in the response? Yeah, I really think it does. I really think it would. I think that looking at responses to smaller components in a bird vocalization would be different depending on the context in which they are in the sequence and what role they play. And I think that would be a beautiful work to do. You don't need EEG or MEG to do that, but you can still ask similar questions. And I think that's where you might get at something that's certainly not word surprisal, but is something that's like word surprisal. That is something like how surprising or unsurprising a given sequence is if it appears in natural bird vocalizations. I think that's a great experiment to do.
Thank you. Any more questions from the audience? I'll ask another one in case people want a bit more time to think about their question. And my other question has to do with distinction between attention and intelligibility. So you're showing how intelligibility modulates these responses in specific ways. Can you, and if so, how can you distinguish the pure effect of intelligibility, so I understand something versus I don't, from secondary effect of I understand something and hence I am more interested in listening to it, so I attend to it more? So do you think what you're seeing is intelligibility or
well, so yes, so I do think what we're seeing is mostly intelligibility. There are certainly some aspects of attention to it. One of the ways that you addressed that question, one of the ways that we addressed the question was to do a control study where we played the same sounds but in a different order. So we played the pre, and then we played the same sound again, another pre, and then we played the clear speech. And so what happens there is that the subject typically, and certainly when asked later in response, paid attention to both because they were slowly learning how to understand this speech. This is 3-band vocoded speech. It can become intelligible with practice, and in the 40 minutes that they were there, it became more intelligible. And so in that case, attention really wasn't so much of a factor, although it can't be ruled out entirely. But we saw this effect disappear entirely.
Thank you, Jonathan. And it's 5 o'clock now, so I think we're formally meant to adjourn. I want to thank Jonathan again for a really excellent and also very didactic talk. And thank you for the audience from all over the world joining in to listen. And thank you to Aero for facilitating. And see you all again next month for the next talk.
Thank you, everybody.
Thanks, everyone. Have a wonderful day.